Well, hello, this is Miss Dagenford, and I wanted to continue our discussion from class about the experiments that created the DNA model. And we started in class with Fred Griffith, and we had looked at how the type of bacteria affected mouse and their ability to fight the pneumonia bacteria. Now, and it had to deal with whether or not it was a smooth form of the bacteria or the rough. And if you recall, um, Fred Griffith and his researchers came up with this idea that there was something that could transform the living R. Now, where we left off is that they were coming up with possibilities. What was the transforming principle? What was the transforming principle? And so they came up with a list, and the list it might seem a little familiar to you because they went through all of the different possibilities: sugars, fats, proteins, RNA, DNA. These are all different types of organic molecules that are in the cell. Now, fats they kind of ruled out from the beginning because fats don't really have that many variations. And so, if you're looking for something that can cause genetic mutations and genetic variability, that wasn't a really good contender. But sugars can have some variation, proteins can have some variations, RNA and DNA. So I want you to think about how an investigator might go about this, because one of these is the transforming principle. We don't know which one. Well, just like a thief who comes into the classroom and steals cookies, we want to eliminate which one is the culprit, which one is the thief. Well, we want to eliminate which one is the transforming principle. So along comes, and you can see the difference in the dates. This took a good number of years, but you can think back in your history days. We had the Great Depression, we have World War II. So you can imagine some of the scientific investigations had slowed down during that time. So along comes Avery, McCarty, and McLeod, and these three researchers were interested in this. They're still trying to figure out which one of these molecules is the genetic material. And so his experiment, the goal was to isolate that chemical that could be that transforming principle, that transformation principle. And so here we have our sugars and proteins, RNA and DNA. Now he came up with a clever idea. Let's just use enzymes, because enzymes, if you recall, enzymes are specific to their substrate. So let's use enzymes to destroy those molecules, and let's see if that transformation from R to S cells still occurred. And this is very similar to how we might figure out who is the thief that stole the cookies. If this keeps happening, if somebody comes in and steals cookies every day from the classroom, and I think it might be, four people, and if every day I just get somebody to distract that person and keep them occupied, and if I come up to my room and say, "Oh, the cookies are gone," well, if that person was being distracted, we know that that person didn't steal the cookies, and so forth and so on until we get to the person who, when they're distracted, the cookies are still there, and then we can go and sit back and go, "Oh, now we know they're the thief." Well, so what we're doing in this experiment is taking away each one to see if transformation still occurred. So up here, I'm just going to put transformation, and I'll put a little question mark. Does it still happen? And so they used enzymes that can break down sugars. And if sugars are the molecule that cause the transformation, if we get rid of them, then we should not see transformation. Well, indeed, we still see transformation. Transformation happened. So sugar is not the transforming principle. Proteins, if we take enzymes that can break down proteins, and remember, if proteins cause transformation and we take them away, then we shouldn't see it. But indeed, we still see transformation. So that means proteins are not the transformation principle. Transforming principle. RNA, we did the same thing with RNA. We used enzymes that are specific to RNA, and we still see transformation. So again, they're able to say RNA is not the transformation principle. And it's only until we get down to DNA. And DNA, if we take it away with enzymes that specifically, my bad, enzymes that specifically are targeting DNA, if we use those enzymes, transformation does not occur. So there was that very excited conclusion that DNA is the transforming principle. And yet there was still this sense of well, wait, is it really? And so people were resistant to the idea that DNA was a genetic material. And so there's one other experiment we want to look at, and it is, and so there is still this question: what is that transforming principle? And so along comes Hershey and Chase. Um, and Hershey and Chase, and we have a man and a woman. I'm forgetting their name. Martha Chase. There we go. And along they come in the 50s, and they use a bacteriophage. Now, a bacteriophage is a virus, and this is the time in history where we're getting a fairly good understanding of viruses now, and so we know what they are. And a bacteriophage is going to be a virus that infects bacteria. And as it infects bacteria, it is a nice way to investigate what gets put into the viruses. Because we knew that viruses infect cells, and so that means they're giving them their information. And so we're trying to use viruses as a nice example, a nice model organism. Now, viruses have very little in their structure. Viruses have a protein coat and a DNA. Well, some of them have a DNA, some of them have RNA, but this one has a DNA molecule inside. Now, the nice thing about viruses, or rather these molecules, my bad, proteins have sulfur, but they don't have phosphorus. And DNA molecules, well, they have phosphorus, but they don't have sulfur. So it gives them a really, really clever way. This is a very elegant experiment. It gives them a really clever way of following these molecules. So if they can use an isotope to label, um, let me get a, there we go. If they can use a label for the protein, and that would be the sulfur. And if they can use, let me get the same color here, if they can use a label for the DNA, and that would be the phosphorus. So just as an environmental scientist might look at grizzly bears, and we're going to tag them with a radio collar and follow them. Well, we're going to use these isotopes that allow us to track, and these isotopes are just a little different. So here's the experiment. They are using protein for one batch of these bacteriophage, and then the other batch has a phosphorus. And remember, those are elements, and because they're elements, most of them have an isotope with it. Now, if you need a quick review, an isotope has extra neutrons, and that means it's got ever so slightly different characteristics. Sometimes isotopes are radioactive, sometimes they're not, but we can oftentimes figure out where they went. Because they're different than the normal molecule. So the first batch has the sulfur in it, and that would be the proteins. So in this flask, we're going to have the bacteria and the viruses. And so this little spaceshipy thing, that's our bacteriophage. And then we have our bacteria. And the second batch is going to be very similar. And we're letting the viruses infect the bacteria. Now, this is going to be where we separate out the protein code of the virus with the DNA within the virus. Now, what they did, they had a time honored technique of separating out the parts of a solution, and that's centrifuging. Oh, I'm sorry. They put it in a blender, and as they put it in the blender, that shakes off the protein coat of the bacteriophage. And then they centrifuge, and the centrifuge is going to separate based on the mass, the weight. If they're heavier, they're going to be pulled down to the bottom. So what we end up having at the bottom of this test tube, this is going to be the bacteria that has those bacterial cells have been pulled down to the bottom of the test tube. Up above is the solution, and so the solution is going to have the protein, and then the bacteria would be down at the bottom. Now, because the protein has been labeled with this isotope, we can tell that the protein has not been injected. So the conclusion of this first batch, the protein is not in the bacteria. Now, the second batch is run the same way, but this time we're going to use the phosphorus that is the isotope. And the phosphorus is going to be made into the new DNA. The DNA is going to be able to be tracked with this isotope. So the same method, the bacteria are mixed with the bacteriophage. The bacteriophage are going to infect. But this time, because the DNA is the the radioactively labeled, the inside of the bacteria now have that. And so if we centrifuge, we'll whirl them around in the blender to shake the protein off. Notice now the proteins are not labeled because we used the labels for the DNA, not for the protein. So the protein, they're not labeled. They're just normal proteins, but the DNA is. And because the DNA is, when we centrifuge it, now the bacteria cells, so the bacterial cells now have the viral DNA. And we know that because it's labeled and we can track it. Well, up here on the fluid, the surrounding solution, there's no labeling. Well, the conclusion, and this, again, this very nice, elegant experiment, the conclusion was simply that the DNA is inside the bacteria. Now, what they could pretty confidently take away from this is that the DNA is the transforming principle. The DNA is what the virus injects into the cell. That's what's important. The DNA is what causes that host cell to be infected. And so Hershey and Chase were really able to figure that out. Well, I hope that helps explain some of the experiments. We're going to be looking at them in class. So come with questions, take notes. I hope you took notes. Please take a moment and go back and annotate your notes, clean it up, and make sure you write down any questions you have. Have a great day.